Great, thank you very much. Thank you for the introduction, and thanks to the organizers. I hope the right screen is being showed now. Is that true? The presentation is? Okay, very good. Yeah, so I'll be talking about our efforts to measure electromechanical coupling in collagen and our attempts to try to tailor, maybe it's a strong word, but try to tailor or control the collagen piezoelectricity by preparing samples with some kind of ordered structure in terms of electromechanical coupling. As we hopefully kind of all know, electromechanics and piezoelectricity is quite a useful property. It's used in actuating and sensing, and as mentioned, in energy harvesting. And maybe what we'd like to do in the future is replace all of these piezoelectric components with biological systems. It might be an interesting thing to do. So we might look to replace the certain materials for some kind of biomedical devices, harness this naturally occurring piezoelectricity that occurs inside of our collagen and other biopolymers within our body. Maybe to use some kind of actuation or MEMS application, maybe all the way up to biomolecular robots. And I imagine that there are researchers working in all of these areas. I'm a bit slow, so we're going to start from a more basic starting point. And so we're going to look at collagen as a prototypical piezoelectric biopolymer. If we can understand this, maybe we can make better progress in going from electromechanics to bioelectromechanics. So the motivation for the work is a while ago, we had looked at collagen and observed, based on some work that had been done a long time ago, that the collagen was piezoelectric. It has this nice diperiodic structure that arises from the organization of the individual collagen monomers. But the collagen monomers themselves have a certain dipole. They have an amine to carboxyl termination, and they all arrange in this kind of ordered structure, and the overlap and gap regions give rise to this periodicity that we see. And it's been discussed that the piezoelectricity might arise from the packing of the collagen molecules within the collagen fibril, and it might have a quasi hexagonal structure if you look at the cross section. So we're curious to understand the origin and biofunctionality of piezoelectricity, the organization of these molecules within fibrils, whether fibrils that become more cross-linked, perhaps with aging or disease, have higher elastic modulus, and maybe different mechanical or electromechanical coupling as well. And what really happens in moisture-rich conditions. We're always looking at things, we're applying a bias when we do our piezo response force microscopy techniques. So we're often looking at dried materials, but what happens? And is the collagen piezoelectricity actually a biofunctional and relevant property? Again, the idea of role of cross-linking in terms of aging and disease, and what happens to our piezoelectric response if we heat the sample, heat the collagen, and undergo protein denaturation. And then finally, can we exploit this piezoelectricity in some kind of application? And to do so, maybe we need to fabricate piezoelectric materials, collagenous piezoelectric materials. Okay, so I should introduce the technique that we primarily use, and it's called piezo response force microscopy. It's a contact mode technique with a conducting cantilever, and we apply an AC signal to the tip. And what we measure is either the deflection of the cantilever owing to the deflection, the expansion or contraction of the material, or we measure the twisting of the cantilever owing to the shear deformation of the material. With the tip in contact, the tip will twist. So we can get somehow vector information or tensorial information about these piezoelectric materials and maybe understand the organization of the structure, organization of the tissues. And so here I show what we call out of plane or vertical PFM. A cartoon of a rat tail tendon is cross section, and we're looking at the end of the fibrils within a rat tail. The rat tail has very highly ordered collagen. All of the collagen is aligned along the length of the tail. And then we can see the cross section of our PFM phase. So we see that some of the fibrils are pointing out of the screen and some are pointing into the screen. And here's a nice example of a collagen fibril that's laying on a substrate. It kind of curves around within the field of view of the image. And when we apply our in-plane or lateral PFM, we're actually looking at that twisting of the cantilever. So we can directly measure shear piezoelectricity. So with a properly oriented sample, in this case with vertical PFM, we might say that we're measuring something proportional to the D33, the longitudinal piezoelectric coefficient. And in this case, we're measuring something proportional to the D15 or shear piezoelectric coefficient. And this image was taken by an intern probably more than 10 years ago, but I really like it because it always highlights this idea of the sensitivity of the technique to the orientation of the fibril. It's the same fibril curved around. And if we look at this PFM phase image, we can see that the amine to carboxyl orientation flips within the image, giving rise to a different phase response from what we see. Okay, and one of the things that we applied this to long ago was cornea, and cornea has a very interesting structure with orthogonal lamella of very small and regularly sized collagen fibrils, and we can find with AFM this location, and it matches kind of quite nicely what we might see with a TEM image of the cornea. So we can see that the fibrils in this location are oriented horizontal in the screen, and the fibrils in this location are sticking out of the screen. So we get a high response in the amplitude in this orientation and a high response in the lateral in-plane signal in this orientation. Okay, and one thing that we've tried to do is reconstruct the piezoelectric tensor of collagen by cutting the tendon at different angles. And in this manner, we could try to recreate the whole piezoelectric tensor. So from this orientation, we could get the D33. From this orientation, then we get the D15. And with this information at different cuts, we're able to obtain D31 and D14 and complete the piezoelectric coefficient, the tensor coefficients. Okay, so back to the idea of changing the humidity, changing the moisture conditions. Here's an individual collagen fibril. In the height image, you can't really see the deperiodicity, but it shows up in the PFM amplitude image here. At relatively low relative humidity, we have a signal that starts to increase as we increase the relative humidity. And then eventually this signal decreases again, and then this system becomes a little bit unstable. So we have an individual isolated collagen fibril. So what we wanted to do was look at collagen within the tendon where it might be a little bit more stable as we change the humidity. Here, the sample is becoming pretty soft. So if we do the same experiment with collagen in the tendon, we can start at this low relative humidity and we see an increase and then a decrease as we keep increasing the relative humidity. I find this interesting. First, the conclusion I'd like to make is that even at these higher relative humidities, we still have electromechanical coupling. So I think that's promising in terms of whether or not the piezoelectricity has some biofunctional relevance. And then also strikingly, I think that between low and higher relative humidities, there's a maxima, there's an increase in the signal. And so this is telling me that at intermediate values, we have some increase in the signal. And so it makes me think about the role of water content in the signal that we measure. Okay, so we also wanted to look at what happens if we cross-link samples. This might be an indication or mimic what happens when we have certain diseases or just get older. So this is a GTA, glutaraldehyde. You can see the PFM without any treatment, and then 5% treatment and 15% treatment. And we have kind of a steady decrease in this electromechanical coupling that we see with these collagen fibrils. We can also do an assay to kind of measure the degree of cross-linking, how many free amino acid groups are available. And we have a reduction in the free amino acid groups when we get up to here, maybe corresponding to a reduction in the piezo response that we see. And so in my kind of simplistic view, I think, you know, as we're increasing the cross-linking, we're increasing the stiffness, and so the fibril can deform less. And so the electromechanical coupling is reduced in that sense. And when we do FTIR and we look at this amide A band, which overlaps with OH stretching, we see a reduction from the collagen control with no treatment, increasing in the reduction as we increase the concentration of the cross-linking. And this is starting to form in my mind this idea of the role of water. So I think this isn't published, we probably have some more work to do, but I think what's happening, and that these studies will kind of fit together, I think what's happening is that the cross-linking is pushing pushing the water out from inside of the fibrils. Crosslinking is taking the place, and so we have a reduced content of water inside the fibril. In parallel with that, in hand with that, if we heat up the tendon, what we expect to happen is that some of the water will leave. And if we heat it up past the denaturation point, the protein will become denatured and the structure will be lost. And so we would have a reduction in the piezo response. So here we have our nice height image. You see all the aligned fibrils with the nice deep periodicity. Here's the amplitude image and the phase image that's telling us that some of the fibrils are up and some of the fibrils are down in the screen. And then as we heat up and we go to denaturation, I think it would happen at 65 to 78. The structure has changed. So we've probably kind of made gelatin at the surface, and the contrast, the signal has reduced significantly. And we see a reduction in the piezoelectric response with increasing temperature. We tried it also for individual isolated collagen fibrils, but I think we didn't quite reach the denaturation temperature. We probably needed to get a little bit higher, but in this case, in any regard, we see a slight increase and then a decrease. The slight increase, I would say, might be due to the initial leaving of water from the surface. And then if we keep increasing the temperature, I think we would see a significant reduction corresponding to the protein being denatured. And we saw this paper now a number of years ago and found it was quite interesting. So in this study, the researchers took fish scales, which have collagen, and they made energy harvesting devices out of them. So basically taking the scales, making a compression of them and kind of slapping them with your hand and generating voltage that way. It's really cool. We had a visiting student and wanted to do something different in the lab. So we got some fish, some different fish. I think that was the type of fish they used in their studies. We got these. And so we took scales from each of them and didn't do a very thorough investigation, but just out of curiosity. So we looked at the PFM and the AFM topography of these samples, just of a few scales of each of the fish. And we can see the nice piezoelectricity. And if I look at the phase, what I see is that, again, fibrils are pointing up and down in some cases. And if I were making some kind of energy harvesting device, naively, I would think that, well, if I had all the fibrils pointing in one direction, maybe I'd get more signal out of it. So if we can start controlling the orientation of the collagen, maybe we can generate even higher charges, higher voltages than are shown here. And interesting for this mullet fish, if we look on the inside or outside of the scale, we saw different structures. And interestingly, I think you can even cleave these fish scales, and they're often transparent. And to me, they bear a striking resemblance to the structure that we saw for cornea, where you end up having well-ordered collagen in certain layers in different directions. And I think that's kind of the cleavage plane when we peel them off. Okay, and a lot of my background has been looking at ferroelectric materials, applying this technique to kind of well-established piezoelectrics and ferroelectrics. And in this field, we're always looking for exotic states and really trying to control the polarization to generate some interesting domain structures. And that's shown in a couple of these examples here, whether it's some kind of vortex domain or some more exotic topological states. And so I kind of raised the question, can we go from this naturally occurring, well-ordered collagen structure where all the fibrils are aligned in one direction, but half of them are pointing down and half of them are pointing up, to something more tailored or more exotic, at least? I mean, ideally, what we would have is have all the collagen, the polar direction aligned in one direction, and that might give us an enhanced electromechanical coupling for energy harvesting. So we tried a number of different ways of preparing collagen. This is collagen that's not forming fibrils. It's just kind of a gel that's then dried. We can see the response. There's some clustering and ordering, maybe under an electric field or something. We could control this a little bit more. So we get this kind of structure. We prepared some samples using like a dielectric focusing effect, so that all of the collagen would kind of arrange or be located at its isoelectric point and then form fibrils. And so here you can see rat tail tendon on the top and our collagen hydrogel that we made. And there is an overall alignment direction in this actual tendon replica that we could then pick up. So we can take reconstituted collagen and make collagen constructs that resemble tendon, although there could be some improvement. We also tried extruding, like you might do in 3D bioprinting. So we tried extruding collagen. We had trouble seeing the collagen fibrils when we did this. And if you look at SEM of an untreated sample, it's hard to make out the edges of the collagen fibrils. If we bleached the sample, if we treated the sample, we could make out some of the collagen fibrils. And with the AFM height image, we couldn't see anything. It's really hard to say what the size of the fibril is, the diameter of the fibril. But if we bleach it and remove some of the non-fibrillar collagen, you can start to make out the sizes and maybe do some measurements. What I found striking was that even if the surface isn't very good in terms of topography, PFM has a way of seeing beneath the surface. And so we could actually do measurements, and we can get a much higher, better degree of alignment compared to the previous example I showed you with the isoelectric focusing. So we can make these fibrils, or these constructs that have aligned collagen, and recreate what we have with our tendons. We did some work with a company called Fibroline, and I found these samples really interesting. They have an overall alignment based on the direction, based on their fabrication technique, which is something like a liquid crystal display technology, a little bit like shearing a liquid crystal during the deposition, and it leads to this overall alignment. The collagen fibrils themselves didn't exhibit deperiodicity, but I did like the fact that they have an overall alignment direction, and this cross structure is something that forms naturally in tendon as well. It's called a crimp structure. And here's our amplitude, and here's our phase. And the striking thing we found in these samples is that, whereas with the tendon, what we saw was one fibril points this way and another fibril points this way. Here, we're actually getting tens of fibrils pointing in the same direction. So it gives us hope for maybe being able to generate a film that has all the fibrils aligned in one direction, pointing in one direction, not just their topographic alignment, but also their polar alignment. So here we have, 10 or 12 fibrils, and so we have a larger area of uniform polarization, which might be useful. Okay, and so what we did was tried to prepare samples that had different fibril size, because that was something that we can control, and we wanted to see if the different fibril size would give rise to different domain sizes, regions of uniform polarization. So we have 500 nanometer fibrils down to these nanofibrils of 100 nanometers. There's still an overall alignment direction that I hope you can see in these images, and the domain structure is a little bit different, okay? And we used a correlation analysis to try to understand the characteristic domain size. And so we can make a link between the fibril size and the characteristic domain size. So for the large fibrils, the domain size in the lateral scale is kind of small. And for the small fibrils, they can actually group together to form a larger region of uniform polarization, but it would require further work, I think, to make the whole film pointing in the same direction. And then just to finish up, what we've tried then is using these films as a substrate to place spheroids and look at cell migration from spheroids that are placed on aligned collagen. At least in this example, these U2OS cells were spreading out in the direction of the alignment of the collagen. We expanded this, so they migrate a little bit more in one direction. We tried to scale this up by incorporating a kind of transfer plate, so we could transfer the spheroids directly to the center of our well plates that had collagen coated on the bottom. And then we can do characterization of all of these different spheroids and look at their migration and automate the analysis. And the thing that remains to be done perhaps is, can we make such a device that also excites the piezoelectricity of the collagen? So this is on a glass bottom substrate, but we could have a plastic bottom substrate, and we could have pressure that would cause a flexing of the well bottoms that might introduce some kind of electromechanical coupling which might further influence the way that the cells interact on the surface. So with that, thank you for your attention. I think the two main things to point out is that we're trying to make these aligned films with controlled polarization direction. Maybe it's not possible, but we're somewhere along the way. And that we can actually make these reconstituted collagen structures that are piezoelectric and mimic the natural structures. And of course, the work was done by the researchers and students and postdocs in the group, supported by a number of funding agencies, and we had a lot of support through various collaborations. But with that, I'd be happy to take any questions.
Thank you so much, Brian. That was really exciting. Thanks for this nice talk. Are there any questions by the audience? So I don't see any questions in the chat, right? So I do have a couple of questions actually, and we do have some time. So please write some more questions so we can ask Brian. So Brian, what are the rules of thumb to have electrically active collagen tissue? So I mean, would it, for example, work with some piece of cartilage? Would that work to get some significant piezoelectricity out of it?
Yeah, so it depends on the location and origin of the cartilage, but cartilage might also have type 2 collagen. There are a number of fibril forming collagens. We haven't investigated that many of them. We have looked at type 2, and we have looked at cartilage itself from like a canine femur. And I can just very quickly show you, this is collagen type 2. So it has a very distinct shape. It's more like a, I don't know, a cigar shape. And it has its angle dependence in the signal as well. In this particular study, we found that the collagen type 1 was a little bit higher response than the collagen type 2. But there are a variety of other collagens worth investigating, because if you just look at one collagen, you're not replicating the ECM in our bodies.
Right. Thank you very much, Brian. So we have a question by Jamie. Jamie, do you want to ask the question? Yeah.
Yeah. Hi, Brian. Is there a biological use to piezoelectricity? Like, is there an evolutionary reason why it might be useful for collagen or a fish scale or a rat tail?
Yeah, it's a problem that's been bothering me for a while. I don't really know the answer to it. I mean, there are a lot of studies that point to, you know, if you look at the way that our bones develop, the collagen piezoelectricity in our bones is a very nice mechanism to help guide cells, because the tissue is always under some kind of strain. And then the strain will generate these positive and negative charges, which might then guide the remodeling of bone and the development of bone. As you know, if astronauts go up into space, their bone density is reduced, and it's that repetitive motion that leads to, perhaps through a piezoelectric mechanism, charge providing some signaling to cells. But it's, I think, much more complicated. And I don't know the evolutionary reason for it, but we know in a lot of cases that charge is important and relevant. It might be like a secondary effect or something along the process, right? The charge might then cause a protein to adhere to that location, which might have a subsequent downstream event. But it's hard to make a really great case that, you know, organisms that exhibited better piezoelectricity were able to adapt to their environment better, and that's why we're here.
Yeah. No, it's really interesting, and it seems possible that piezoelectricity has a use. It's just we haven't known about it long enough,
or it could just
haven't
be
had a
that
way of looking at it.
Yeah, or that it's everywhere. It's in everything, and as long as the crystal structure allows for it, so it might just be a byproduct of that organization. But it might, yeah, and that might arise because of the self-assembly of those materials. So anything that has a dipole has a chance of being piezoelectric, and maybe it's because of the dipoles that they arrange in the way that gives them the overall macroscopic piezoelectric property. But there might not be a biofunctional relevance for it. But certainly there are studies where people are making piezoelectric biomaterials and using it for wound healing or something, and showing that the tissues heal faster or the cells proliferate more because of the presence of charge.
Thank you.
That sounds really great, Brian. A lot of potential for that. Yeah. Are there any further questions? So we would have time for another final question.
So there is a question from one of the contributed speakers, Andreas. Maybe you can unmute yourself and
definitely
ask yourself.
Yes, I just wanted to ask whether the align substrate that you used for the spheroids, did you achieve also to put spheroids in 3D collagen gels, align collagen gels, or it was a 2D study?
Yeah, it's a great question. It's just a 2D study. So it's somewhere in between 3D cell biology, and so it's like bridging the gap, because if we do a lot of studies on a glass slide and you put cells on a glass slide, we don't really have flat glass surfaces in our body. So building up in that direction, it has a certain topography, it has a certain roughness, but we still call it 2D. It's 2.5D maybe.
Okay.
But they're thin films. Thin films.
So, and you were watching the cells that are attached of the line.
Yeah, we were
substrate.
watching the cells disseminate from the spheroid and spread out on the collagen. So the collagen provides not just the topographic and surface chemistry that the cells might expect, but also the modulus, which we also measured with AFM.
Thank you. Thank you very much.
So thank you very much for the great talk, the great questions.